controller for you, sir. Thank you.
G'day everyone, and thank you very much for the introduction and also for the invitation to be able to present to you today. I think it complements well with the other presentations that we've heard today, particularly in those in terms of circadian rhythms and just more recently sleep. But we will be sort of switching gears a little bit because, again, the type of work that we do is sort of more sort of basic research or preclinical type of research to get sort of real insights in terms of the role of the brain in control of feeding and blood sugars. So for full disclosure, we have had some research support from Novo Nordisk to support some of the work that we've been doing. So as I mentioned, our work focuses on the role of the brain in the control of energy balance and also blood sugars, and determines whether defects in this control system contribute to the pathogenesis of obesity as well as diabetes. So the outline to go over today is that, first of all, we're going to look in terms of how energy balance is regulated. We're going to look at in terms of the brain and sort of what neurocircuits are involved in the control of feeding, as well as metabolism and blood sugars. Looking at the role of the brain in the control of glycemic control, and then whether the brain can actually be a target for obesity and or diabetes treatment. So the incidence and prevalence of diabetes is increasing throughout the world. It's estimated that up to 2 billion people are classified as overweight and more than 500 million as obese. And this is obviously similar trends are seen in the US. Some of you may be familiar with this data from the CDC looking sort of at the obesity rates just over the last few decades. And so this is defined as in terms of obesity of a body mass index. And again, it's not perfect, but it's a good indication for sort of large population studies. And so you can sort of pick your favourite state as we sort of walk through here in terms of, from the 1980s, as we go through into the 1990s, you can see about sort of 10 to 15% of people being classified by obesity. As we start to go into the 1990s, we start to see that increases from sort of 10 to 15%, and then getting up towards 20% by the time we get to the turn of the century. By now, it's now starting to escalate from that sort of 10, 15 to 15 to 20%, and now getting up to about 25%. You can sort of see Colorado's just hanging on here for a little bit longer, but unfortunately that sort of starts to go to the next level as well. And as you look in terms of the last 10 years, then it continues to increase from this 20 to 25, to greater than 30% over this period of time. And so again, this is a really rapid sort of time frame when we start to see these escalating rates. So if you take a snapshot over the last 30 years, you can see, from the early 1990s, sort of got 10 to 14%, and jumps up to about 20% the next decade, 25%, and now sort of the stage where it's sort of most of the country is over 30%. So similar trends in terms of both the incidence and prevalence is also occurring in terms of type 2 diabetes. Here we're looking at both the incidence and the number of people with diabetes, and you can sort of see there's a low, steady increase trajectory since the sort of 1960s, but this has sort of really escalated a lot more over the last 20 years, and it's estimated these days is that nearly up to 10% of the population in the US has diabetes, and then it's approaching about 30 million people. And as you can see, it's not going in the right direction. It's likely to get worse before it gets better. And the reason why this is important because, again, obesity and diabetes has sort of increased with a lot of medical complications, including both macro and microvascular disease. As we discussed today, really affects every organ in the body, increasing the risk of renal, pulmonary, coronary heart disease, stroke, blindness, certain types of cancer, and even sort of skin infections sort of leading to amputations. And so this obviously has an enormous, health cost and financial costs related to it. But unfortunately, it also increases sort of mortality. And so you can see here in terms of sort of the risk of death, as the increasing level of obesity, there's a marked increase. So the question is, how have we got here? Particularly in terms of, this is over a pretty short period of time. We're just talking 30 or 40 years. And obviously our genetics really haven't changed that much over this period of time. And so when you think about it though, in terms of energy balance equation, in terms from a math side of thing, it seems pretty easy in terms of, yes, it's a balance of energy intake and energy expenditure. So it's simply explained is that obviously if you consume more calories than you're expending, you're going to gain weight, but more of a case, well, it's easy to lose weight. It's just simple. You just eat less, exercise more, and you're going to sort of lose weight and keep that weight off. But it's a little bit more sort of complex than that. And so that's obviously in terms of, the simplicity of it is obviously sold in sort of every sort of media avenue that you can look at. And there's always the new latest fad diet or supplement or something you take to be able to sort of melt the pounds away, to be able to lose that, 15 or 20 pounds or however you want to look at it. And so, again, a lot of these will work. It's actually not too difficult for most people to lose weight. What the real challenge is to be able to keep the weight off. Which then raises this question in terms of, how is body weight regulated? And so, because we know is that in terms of the number of calories we may consume on any given day, that varies tremendously. So particularly coming up to the holiday period, Thanksgiving and Christmas, you eat enormous numbers of calories, yet other days you barely eat anything at all. So how does the body know? Well, it does it through a process known as energy homeostasis. This is defined as a physiological or sort of biological process where energy intake is matched to energy expenditure to promote stability of weight or the amount of body adiposity that you have. And the way the concept is thought to work is through an adipostat theory. So similar to a thermostat in your house. So probably similar to Seattle up here, it's like winter is coming. So similar in terms of thermostat is that obviously the room's colder, it senses that and then pumps out more heat to be able to maintain the temperature in the room. So it's thought to act in a similar type of manner so that you're at a certain level of weight and then you may be in negative energy balance and you're losing that weight. Well, the body recognises that. And what it does, it engages adaptive responses not only to limit further weight, but also to be able to restore the weight back to where it originally was. And obviously we can feel and sense some of that, particularly in terms of the drive to eat, the hunger. But also in addition to that is that your body tries to conserve its energy through that period of time to, again, limit the amount of weight you're losing and to be able to restore it back to normal. So that's sometimes even more the frustration when people are dieting. It's like, I'm actually eating less calories than I'm due, but I'm still not losing any more weight. Well, part of that reason is your body's tried to protect itself. So unfortunately for most lifestyle or behaviour or exercise or modification programs is that most people unfortunately regain the weight like 80% over a 5-year period. And Kevin Hall's done some recent work at the NIH showing that's indeed the case. But in addition to protecting against weight loss is that the same thing happens in the body, tries to protect against weight gain during positive energy balance to be able to sort of limit how much weight you regain, but also bring it back down to where it began. And obviously part of that is feeling of fullness as well as increasing your energy expenditure. And a couple of studies to be able to sort of support this. One was a Vermont study of obesity, which actually was done in prisoners in the late 60s, early 70s. So this is just an example of sort of one sort of particular individual eating about sort of 1,200-1,300 kilocalories, and they gave them incentives to be able to increase the caloric intake. And so you can see that here they're eating about 4,500-5,000 kilocalories, so 3 to 4 times the amount. So that's expect that the weight gain comes up. What you see interesting after a few months, the weight gain doesn't just continue to go up and up and up. It actually sort of then stabilises. It sort of starts to defend an elevated level of weight. And this went for about 6 months, and obviously they took everything away. But the interesting thing is that basically they hardly ate at all for the ensuing sort of days, weeks and months. And you can see the weight actually comes back down to where it originally started from. So that these adaptive responses, importantly, not only occur in sort of normal weight individuals, but also in the setting of obesity as well. This is some work done from Rudy Leibel at Columbia, where he took a range of individuals from normal weight to obese, and then looked at the metabolic response to either weight gain, where it was 10% increased, or weight loss. So here we're looking at the effect in terms of energy expenditure over fat-free mass. And so the open squares as sort of the initial weight, and you can see for those individuals that gained weight, they're all above the line, indicating they had an increase of energy expenditure. So what you might expect, conversely, is again we've got all the individuals at normal weight. When you look at weight loss, they're all below the line, indicating a reduction of energy expenditure. And again, this sort of a dose response as well. So those that lost 20% of weight even fall further below, indicating an adaptive response to be able to reduce energy expenditure during weight loss. So a few key take-home messages from this. It suggests, one, there's biological mechanisms that protect both against weight gain and weight loss. They occur in both normal weight as well as obese humans. That obesity involves the defence of elevated body weight rather than just any absence of regulation. And we'd hypothesise it's a disorder of the energy homeostasis system rather than just, for obesity, a passive accumulation of weight. So in terms of how does this occur, and sort of the overarching hypothesis is that body weight's regulated by a sort of a classic negative feedback loop. It was originally hypothesised that there was some type of signal which was released from fat, which would just communicate to the brain in terms of what the energy stores were. The brain would then sense that information and accordingly sort of regulate systems that affect both food intake and energy expenditure to maintain body fat stores. And there was great excitement in the obesity field in 1994 with Jeff Friedman with the discovery of the OB gene, which is exclusively expressed in adipose tissue, which produced the hormone leptin. And so this is a hormone which is secreted in direct proportion body fat. It enters the CNS, the brain, in proportion to its body adiposity levels, and then signals in key brain areas to be able to reduce food intake, stimulate energy expenditure, and sort of induce weight loss. This sort of also fits in with the overall mechanism in terms of conditions of negative energy balance, or sort of such as going on a sort of a diet or caloric restriction. In this setting, as a result, there's a reduction of body fat stores, and consequently there's reduced leptin levels. And so you're sort of turning off the brake, because again, normally this is activating pathways that reduce food intake. And so by reducing the leptin levels, consequently it's sort of both promoting an increase of food intake and a reduction of energy expenditure. But the real proof in the pudding in terms of this is critical in the regulation of energy balance was the consequence of leptin deficiency, because then there was no signal to be able to really tell you to stop eating, and that was obviously shown here in the OB leptin deficient mouse. So again, characterised by both an increase of food intake, reduced energy expenditure, and as you can see, massive obesity. But this was not only found in mouse models, but it was also found in humans as well. And so this is an individual with leptin deficiency, 3 years old, weighs nearly 100 pounds. But the most exciting aspect was if you administer leptin back to these individuals, similar to what we saw with the rodent models, is that essentially you normalise their body weight and every neuroendocrine axis in the individual. So there's great excitement at the time, is this the magic pill that we've all been searching for all these years for the treatment of obesity? Well, there's only a handful of humans with genetic leptin deficiency. The most common forms of human obesity aren't really caused by a deficiency of leptin. As I made mention, it's secreted in direct proportion to body fat. So for obese individuals actually have higher levels of leptin. And so it's sort of a case the leptin's not working as well, which has been termed like a leptin resistance. Now, while the underlying mechanism by which that occurs still remains to be elucidated, there's been a lot of work done to be able to try and understand how and where leptin works in the brain to be able to mediate its effects. So one of the main questions in the field is, what is the central mechanism whereby leptin regulates energy balance? So one particular area which is implicated is that in the hypothalamus. This is based on sort of classic lesioning and stimulation studies more than 50 years ago, showing how potent effects to either increase food intake or cause starvation. And so here, for those not familiar with in terms of brain anatomy, we have a sort of a sagittal slice up top, and then we have sort of a coronal slice. We have the hippocampus, the thalamus. Down the bottom here, we have the hypothalamus. And within that, sort of a number of discrete nuclei. And the little box there, you can see the little bell trumpet shape, that's the third ventricle. And one particular area which has been of interest is that area known as the arcuate nucleus. So the first work was to be able to sort of determine where the receptor may be expressed in the brain. And this is work by Dennis Baskin at the University of Washington using in situ hybridization. And you can see the leptin receptor is abundantly expressed in the thalamus, but particularly there, you can see it there in the arcuate nucleus. Now, this is also an area which is well known to be able to sort of express a number of neuropeptides which are important in the regulation of feeding and metabolism. And of those is, again, we've got a blow up here of the sort of arcuate nucleus with the third ventricle. In green, we have staining for neuropeptide Y. So this is one of the most sort of potent orexigenic effectors to be able to stimulate food intake. Now, adjacent to these are cells, they also express leptin receptor here in red that express POMC. Now, these do the opposite effect. When these are activated, they potently reduce food intake. So the third player to the field is ones which express agouti-related protein, or AGRP. So as we'll see, these are co-localized with NPY, but they also block the actions of the melanocortin system. So again, you can see here in red as those cells expressing agouti-related protein. Now, the only place in the body these are expressed is just in this particular area within the brain. You can see they're co-localized with that, expressing NPY. There are several lines of evidence suggest these are part of the melanocortin pathway, and they play a critical role in the regulation of energy balance. And they're regulated both sort of by neural, hormonal, and nutrient-related input. So based on this, we've sort of put together an overarching model in terms of the melanocortin neurons, or those POMC neurons shown here in green, where they're sort of activated by these adiposity signals, insulin and leptin. Then this releases a neurotransmitter, alpha MSH, which then binds on a downstream melanocortin receptor, and when activated, it potently reduces food intake, increases energy expenditure, and causes weight loss. Conversely, next door to that is the NPY-AGRP neuron, and these are inhibited by both leptin and insulin under normal circumstances, where they're stimulated by the stomach-derived hormone ghrelin, and when these are activated, they potently increase food intake, and they can do it through a number of mechanisms. So one of these is it can release NPY on its downstream receptor, but second of all, it can release AGRP, which blocks the signal on the downstream melanocortin-4 receptor. But third, it also sends GABAergic projections to different brain areas, including to this POMC neuron. So in essence, if you sort of the analogy to driving a car in relation to feeding, you're both putting your foot onto the accelerator, but you're taking your foot off the brake at the same time. These are obviously activated during conditions of negative energy balance, which can explain in terms of why under those conditions it's hard to be able to maintain that weight loss because these neurons are activated. So this model sort of really stood the test of time over the last sort of 20 years or so in terms of how these neurons are regulated by inputs like insulin and leptin and ghrelin, but it's sort of evolved over more recent years. And that in addition to feedback control, it's also regulated by feed-forward control, which promotes energy homeostasis in a manner which is sort of anticipatory. It's predicting future need rather than responding to a change in a variable such as a change in body fat. And it's similar to what happens with other homeostatic systems as well, where responses are engaged before there's a drop in a variable. So among the sort of feed-forward sensory cues for AGRP neurons includes the sight, the smell, the taste of food. They're also inhibited by sort of gut-derived satiety signals like cholecystokinin and GLP-1 as well. But they're also regulated by other sort of external variables, one including circadian rhythms, but particularly by changes of ambient temperature. And there's real growing evidence is that systems regulating energy homeostasis are tightly coupled to thermoregulation. And so, because cold exposure is one physiological challenge which can have a pronounced effect on the energy homeostasis system. So during cold exposure, there's a marked increase of energy expenditure, and that's required to help maintain body core temperature and to be able to avert hypothermia. So this increase of energy expenditure must be also offset by an increase of energy intake in order to maintain energy balance. And this can be critical for survival because obviously a failure to do so actually runs a risk of a progressive loss in body fat stores. Now, while lots known in terms of the neurocircuitry involved by which sort of cold increases energy expenditure, less is really known in terms of how it actually affects food intake. So our overarching hypothesis was that activation of these AGRP neurons plays an important role in the adaptive increase of food intake during cold exposure. And this work was done by a talented postdoctoral and then faculty member at the University of Washington, Jennifer Dean. So the first question we wanted to simply ask is, how rapid do we see these effects on both energy intake and energy expenditure in response to cold exposure? So to do this, we use rodent models, just regular sort of wild-type mice. And so then we place them in an indirect calorimeter so we can make continuous measures of both feeding and energy expenditure when the animals were either at 14 degrees, relatively mild cold exposure relative to room temperature 22 degrees. And what you can see here relative to 22 degrees in green is basically you see virtually an instantaneous increase of energy expenditure in the animals subjected to 14 degrees. And again, this sort of makes sense. If I walk straight outside at the moment, is I want my thermogenic machinery to start kicking in straight away. I'll try to keep warm. Not sort of coming in an hour or two later until I've had a drop in core temperature. And you can obviously see this is increased throughout the duration of the exposure. And here we went over a 24-hour period of time. But what's interesting to us is that somewhat similar to the increase of energy expenditure, we also saw a rapid increase of energy intake that also occurred. And again, this happened over the 24-hour period of cold exposure. So these data suggest that an increase in energy expenditure and energy intake during cold exposure are sort of rapid in onset and suggests the mere sort of perception of cold is sufficient to increase both of these and occurs based on the time frame independent of a negative feedback signal which may obviously be recruited at a subsequent time. We then wanted to address whether cold exposure activates AGRP neurons. And to this end, we collaborated with an investigator at the University of Washington, Richard Parmenter, who generated a mouse model. And so he did this in terms of he had an AGRP which had a fluorescent marker on it, green fluorescence protein. So every AGRP cell was green. And so then we had animals relative to 22 degrees. And we either put them in a warm environment of 30, which is sort of ambient, approximate ambient temperature, and then 14 degrees, a mild cold exposure. Sacrifice the animals 90 minutes later, and then use immunohistochemistry to look at FOS, which is sort of a marker of neuronal activity. So again, here we have sort of a blow-up of the cartoon of the hypothalamus. We're focused right here around the third ventricle and the arcuate nucleus. You can see those green cells expressing GFP are the ones that express AGRP, and in red we have the FOS. So again, a thermoneutral environment at 30 degrees. You see relative to 22 degrees and then 14 degrees. You can see both the number and sort of percentage, particularly as you can see the yellow colour of a co-localisation of the FOS and the AGRP neurons. So we quantitated this, and you can see there's very little at 30 degrees, but you can see particularly relative to 22 and 14 degrees, we have not only increased number, but the percentage of AGRP cells that express FOS. So notice here it's about 20-25% of those cells. So thus acute mild cold exposure activates, but it's only really a subset of these AGRP neurons. I said about 20-25%. Because in contrast, if you do fasting or ghrelin, probably 70-80% of the AGRP neurons light up, which sort of raises the possibility is that the subset of neurons activated by cold are important in the regulation of feeding, but other AGRP neurons that are not may be important for other behavioural metabolic functions of these particular neurons. So FOSs are good, but it's just a snapshot in time. We wanted to really know how rapidly does cold exposure activate these particular neurons, because the feeding response happens early. We'd expect these neurons to basically start being activated at a time either before or similar, not sort of an hour or two later. And so to do this, there's been some real pioneering breakthroughs in the neuroscience field. Seems some of the stuff you can do now is science fiction, even from 10 years ago. And so this is a technique called fibre photometry, which allows you to measure the activity of specific neurons in the brain in live conscious animals. So to be able to do this, there's sort of 3 or 4 key ingredients that you need. For one is that there's been growth in terms of mouse genetics. So you can get a particular mouse generated which has a Cree driver to your favourite receptor or neuropeptide or protein. So for us, we're focusing on AGRP, so we had an AGRP Cree animal. The advantage then is you can then get a viral construct which is generated, which is Cree dependent. So the only cell it's going to hit is the one that expresses Cree. It doesn't affect any other cell. And so it takes advantage of using a sort of calcium indicator virus. So when a neuron turns on, calcium goes up. And so it can actually then do that by it. And what you do is you implant like an optic fibre just above or where you do the injection. And so you let the animal recover, sort of train it up to a task for a few weeks, and then you conduct an experiment. And so you can then do the challenge, whether you present it with food, if you put it sort of, obviously for us, challenge it in a cold environment. You can give an injection and then be able to actually have a look in terms of whether the neurons turned on or the neurons turned off. So for us, we had our AGRP Cree animal. We gave our Cree-dependent calcium virus. You can see in that middle panel, the only place you can see it is in those AGRP cells. And then we waited for a few weeks, and then we first tested the animal by fasting them overnight and then just presenting food, and that's shown to rapidly inhibit the AGRP neuron. So in those animals we then conducted our study where we then hooked them up to the sort of recording system, and then they were in a cage where they had a plate which you could change the temperature on them so they could either be exposed to a cold or sort of a warm environment. And so this is what we saw. So the first one is looking at a heat map. And if it's dark blue, it means the neurons are quiet. If it's white, it means they're firing a lot. And as you can see, when you can see the animals at 30 degrees, you can transition the animals, is that there's an increased activity. This is quantitated in the middle panel, looking at sort of at the time profile. You can see them at 30 degrees. It takes about 60 seconds for the ramp to go to 14 degrees. You can see a marked increase of the activity of these neurons as it's quantitated here. So these suggest that these neurons are rapidly activated in response to cold exposure. Again, this is unlikely through a feedback mechanism, but it suggests of a feed-forward mechanism. But ultimately, we want to test the most important physiological question is whether the activation of the neurons is required for cold-induced hyperphagia. And again, this is another neuroscience tool that we can use, which uses some of the same principles. Again, you can have your favourite Cree driver mouse, and here we have a sort of a viral construct which is engineered where it can be either activate the neuron or it can inhibit the neuron, but it's only in the presence when you give a ligand, the CNO, relative to a saline control. And the advantage is you can use animals as their own control with a randomised crossover manner. So again, here we had an AGIP Cree animal. This time we gave a bilateral injection of the inhibition virus, and you can see they're expressed there in the arcuate nucleus. So the hypothesis is that if we have these animals and then we're going to place them either at 30, 22 or 14 degrees, and that each of them are either going to get CNO to inhibit the neuron or a vehicle. So the prediction is that at 30 degrees it's not going to do much because I just showed you the neurons really aren't active at that temperature. But it would have an effect when the animals are exposed to cold. So consistent with that, when you look at the food intake, again, relative to 22 degrees, the food intake is reduced. It's increased at 14 degrees. And you can see in the same animals when we gave CNO, it blocked the effect of cold to be able to increase food intake. Now this is selective for food intake and not energy expenditure. Again, you can see relative to 22 degrees, energy expenditure is reduced at a thermoneutral environment. It's increased with cold exposure, but when we give CNO to inhibit the neurons, everything sort of remains intact. So this suggests that AGIP neurons are required for cold-induced hyperphagia, but not cold-induced increases in energy expenditure. Now, first pass, that may seem a little bit confusing because I said fasting activates AGIP neurons. That increases food intake, but it reduces energy expenditure. Whereas in cold exposure activates AGIP neurons, and you both increase food intake, but you also have increased energy expenditure. And so to us, it suggests that AGIP neurons are not just all one and the same. Rather than being homogeneous, there's heterogeneous. And there's functionally distinct sets of AGIP neurons that exist. And that the ones that are involved in cold exposure are important in the regulation of food intake, but they're distinct from other AGIP neurons which are important for other sort of metabolic and behavioural functions. And we know in terms of AGIP neurons, they have a wide variety of sort of metabolic and behavioural effects. Obviously not only feeding and thermogenesis, in terms of controls of blood sugar, reproduction, social behaviour. As I made mention, they're regulated by a lot of various different types of hormonal, nutrient and sensory related input. And so again, the hypothesis is that in terms of cold exposure, it's only activating sort of 20-25% of these neurons. They're the ones important in feeding. Where in contrast to fasting or growling, the majority of these neurons are activated. And so while some of those neurons may obviously be important in feeding, it's activating different populations of neurons which are mediating these other effects. And current work is ongoing to be able to try and sort of really distinguish between these and try and understand where they project to in terms of different downstream brain areas to mediate these effects. So the next question in terms of what happens in the set of sort of high-fat diet-induced obesity. There's some work done from both Zach Knight as well as Michael Krashus we've shown that in setting of high-fat diet feeding, it blunts the AGRP neuronal as well as a behavioural response to a number of different things, not only the sensory detection of food, in terms of administration of sort of macronutrients into the gut, but also sort of gut-derived satiety peptides such as CCK and ghrelin. And I won't go through all the details, but we also show this is also similar to the effects on cold exposure. So essentially when you repeat these studies and you do it in an animal that's been on a nice tasty diet for 3 months, is you put them in the cold, they still show a nice robust increase of energy expenditure. But when you look at the food intake, there's no increase of food intake at all relative to animals at 22 degrees. And then similar when you look at the activation of the neurons while they're activated, instead of 20-25%, it's only like 5-8%. So again, suggesting is that in that particular setting, there's the ability of the neurons to be able to increase food intake and be activated by cold is blunted in a set of high-fat diet feeding. So to put this in the context of obesity, again, when these neurons are turned on, we know that they sort of stimulate food intake and sort of conserve energy. And so if they're not receiving this inhibitory input from the nutrient and hormonal inputs from the body, it raises the possibility is that their activity actually increases, and whether that contributes to the pathogenesis of obesity are things that we are looking forward to be able to continue to work on. So this is looked at in terms of the role of the brain in the control of energy balance, but we're also interested in the role and the control of blood sugars, which isn't exactly new. It was actually sort of first proposed from Claude Bernard 150 years ago when he demonstrated if you puncture the floor of the fourth ventricle, you actually induce a diabetes phenotype. But since the discovery of insulin about 100 years ago, really all the work on sort of diabetes is really focused on insulin in terms of secretion and obviously its action in peripheral tissues. So as we know, you ingest a meal, it's absorbed in the gut, and together with sort of incretins, these act on the beta cells in the pancreas to secrete insulin, circulates around and acts on peripheral tissues such as the liver to suppress glucose production and muscle to increase glucose uptake to be able to lower blood glucose. And obviously there's unequivocal evidence in terms of the pathogenesis of type 2 diabetes results in not only sort of reduced secretion, but reduced sensitivity of insulin in these peripheral tissues, leading to a rise of blood sugars. So in other words, the pathogenesis of type 2 diabetes involves sort of a failure for pancreatic beta cells to compensate, to be able to further increase insulin in the setting of severe obesity-associated insulin resistance. And as a result, most of the therapies that we've heard about today for type 2 diabetes is really focused on insulin secretagogues or insulin itself to increase insulin secretion, insulin sensitizers, although over recent times there's been development of other treatments like the SGLT2 inhibitors to be able to treat diabetes. But these are all sort of lower blood sugars, but as soon as you stop treatment, blood sugars all come back up. So there's lower blood sugars. So it treats diabetes but doesn't necessarily sort of cure diabetes. So we hypothesize is that the brain works with the beta cell in terms of controlling blood sugar. The brain can obviously sense nutrients like glucose, fatty acids, various hormones, leptin, GLP-1, fibroblast growth factors, and can obviously, one, talk to the beta cell to be able to manipulate insulin secretion, but also talk to the peripheral tissues as well in terms of regulating blood sugars. And again, most definitive evidence that the brain can play a role goes back to our friend the OB mouse. It's not only associated with obesity, but it's also associated with insulin resistance and type 2 diabetes. And again, if you give leptin back to these animals, or obviously as well as humans, you normalise these particular responses. But if you give tiny doses which have no effect on food intake and body weight, you still have profound effects to be able to improve glucose metabolism. So not only are there genetic models of leptin deficiency, there's also acquired models as well, which occur independent of obesity. And this is sort of known as lipodystrophy. And so these are conditions where you have very low leptin levels because you don't really have much fat around at all. So what happens, rather than being stored in adipose tissue, it goes where places you don't want, like liver and muscle. And so this is associated with severe insulin resistance and diabetes as well. And giving back a leptin, whether it's a rodent or sort of humans with lipodystrophy, it essentially cures their diabetes. And then from rodent models, it also suggests that the brain is involved, because if you give tiny doses in the brain, you also cure their diabetes. Now, another acquired model is actually what happens in type 1 diabetes. So type 2 is about 90% of all diabetic cases. It's about 10% for type 1. As we know, it's caused by autoimmune destruction of those insulin-secreting beta cells. And obviously, with no insulin around, you have increased rates of glucose production, reduced glucose uptake, and elevated blood glucose levels. In addition, is without insulin around, you can't really store any fuel into adipose tissue. Now, I know insulin's a cornerstone of therapy for type 1 diabetes, and work for the DCCT has really shown is that not only for intensive insulin therapy reduces diabetic-related complications, but it's also obviously increases the risk of hypoglycemia, but also increases weight gain as well. And obviously with that insulin, you now have the ability to be able to store fuel in adipose tissue. And so that's not just a storage organ, but it's also an adipokine organ as well. And so again, in terms of type 1 diabetes with no or little adipose tissue, is that you don't have any leptin as well, being a condition of both insulin and leptin deficiency. So given that we know that leptin plays an important role in feeding and blood sugars, we're interested in what happens if we give leptin back to the brain in a rodent model of type 1 diabetes. So is leptin action in the brain sufficient to lower blood glucose in a rodent model of type 1 diabetes? So to do this, we had a rat model, and here we implanted a cannula to the lateral ventricle. And these animals got vehicle or a chemical, streptozotocin, which causes destruction of the beta cells to cause diabetes. And then we implanted an osmotic mini pump, which contained either vehicle or leptin, and connected to the cannula to be able to do an infusion directly into the brain. So you can have a look, first of all, at insulin leptin levels over time. You can see after we give the streptozotocin, there's a marked reduction in both insulin and leptin levels relative to non-diabetic animals. And that with leptin, there's no effect, particularly in the leptin, there's no effect to be able to increasing leptin levels in the circulation. Again, if you look at blood sugars, in this model, they become hypoglycemic within 24 hours relative to the non-diabetic controls. Again, in the leptin-treated animals, first of all, we waited for them to develop the hypoglycemia and then started treating on day 3. You can see with the leptin infusion, it normalizes blood glucose. So this is giving a tiny dose of leptin directly into the brain of a rodent model of type 1 with no insulin, and we have a powerful effect to be able to normalize blood sugar. Can't be explained by its effects to reduce food intake. It did modestly do it, but can't explain that effect to normalise. And it can't be explained by leaking into the periphery because there was no effect subcutaneously. So thus, continuous infusion of leptin completely normalises blood glucose in a rodent model of uncontrolled insulin-deficient diabetes. We then went to see, well, how does this occur? Obviously, it's not due to reduced food intake. Can't be explained by glycosuria. There's no glucose at all in the urine. There's no beta cell recovery. Even if you give glucose, there's no effect to be able to increase insulin levels. We did tracer studies to see where the glucose went, and there's a marked increase in peripheral tissues like muscle, and also potently suppressed glucose production. So this really provides evidence that the brain has the inherent capacity to normalise diabetic hypoglycemia. And obviously we heard some talks earlier today in terms of therapeutic implications whether supplementing insulin with leptin may be a useful adjunct in terms of the management of type 1 diabetes. There was sort of a short-term study to be able to look at this. There was some beneficial effects. Most of it might have been sort of associated with the weight loss. But again, the real sort of limiting, as we know, in terms of type 1 is a lot of them are now obese. And so in obese individuals, leptin doesn't really work as well. So we'll see in terms if this moves forward or not. We then wanted to look in terms of the underlying mechanism by this was done. And this is done by a postdoctoral fellow, Tom Meek, and it sort of goes back in terms of what happens in the brain. And as we said, similar to fasting and OB mouse, in this model you have both increased levels of NPY and AGRP, reduced levels of POMC, and these are all normalised by the leptin treatment, suggesting that the melanocortin pathway may be important in these effects. And so we asked whether that was indeed the case, where the antibiotic effects in this model require melanocortin signalling. So to address this, we turned to our model. This time we sort of gave 2 bilateral cannulas, implanted them. The animals were then given streptozotocin to induce diabetes. And then we one gave vehicle leptin, and the second one we used, again, a pharmacology approach, where we gave a melanocortin receptor antagonist. So here we're looking both at the food intake. You can see in the diabetic animals is that they have this diabetic hyperphagia, this increase of food intake, obviously hyperglycemic as well. Again, consistent with what we've shown before with the leptin, we normalise the food intake, we normalise the blood sugar, but when we give the melanocortin antagonist, it fails to blunt the effect of leptin to normalise the blood sugar. So that suggests that leptin's effects to normalise diabetic hyperphagia and hyperglycemia requires melanocortin signalling. Now again, as I may mention, is that in terms of type 2, leptin's really not going to work as well so much because of the so-called leptin resistance. But can the brain actually be a target in terms of diabetes treatment in models of type 2 diabetes? Now over the years, there's been a lot of interest in terms of fibroblast growth factors. These are quite often involved in development and differentiation and survival, but there's been real emphasis on the endocrine FGFs, including FGF19 and FGF21. And we and others have done other work to suggest a lot of those effects are mediated by the brain. We're also interested in this peptide FGF1, mainly from work that Ron Evans did, where he gave an administration, a single dose of the FGF1 subcutaneously, and you can see there, this is in an OB mouse. Now the effects are quite long here in terms of the effect to be able to lower blood sugars, over sort of up to a 48-hour period. So we then wanted to be able to ingest whether FGF1 action in the brain is sufficient to lower blood glucose in this rodent model of type 2 diabetes, and this was done by Jared Scarlett, a paediatric gastroenterologist who's now on faculty at UW. So again, we followed a similar approach. This is a obese diabetic OB animal. Blood sugars at 300. Planted with a cannula. This time we just gave 1 single injection of the FGF1. You can see in the vehicle they stay around 300, and the FGF1 it came back down. And normally, sort of when you look at this type of graph, it's sort of like maybe it's over a period of minutes or hours. Amazing thing was this was over weeks. So a single injection to an animal dropped the blood sugar to normal and it stayed there. And it defended that blood sugar. You gave glucose, went up, comes back down to that same level. Give insulin, goes down, comes back down to normal. Now, while there's a transient effect to be able to reduce food intake, this effect can't be explained by any changes of food intake or body weight. More importantly, it's not associated with hyperglycemia because we did it in wild-type animals and don't see it. So this is sort of unprecedented in terms of what we've seen before. And we want to be able to try and understand how on earth does this work. So we used an unbiased approach, initially using transcriptomics, then we did gene expression electrophys, and all pointed back to some of the work we did before. It was associated with the inhibition of these NPY AGRP neurons. Probably most impressive effect was that 2 weeks after the injection, we then electrophysically looked at their neurons, and sort of normally you have wild-type animals that markedly elevated in terms of the activity of the OBOB animals. This is back down to sort of normal levels. We also showed that this also requires a melanocortin pathway. And so together it sort of suggests whether increased activity of these particular neurons maybe, obviously one associated with diabetic hyperphagia, but maybe also contributes to diabetic hyperglycemia, because part of the effect of the FGF1 to be able to induce this diabetes remission occurs in part by inhibiting these particular neurons. So hopefully today I've been able to sort of tell you a story in terms of, yes, the brain does really play an important role in not only the regulation of energy balance, but also plays a role in the control of blood sugars as well. And that, is that, potentially disorders or the ability to be able to sense or respond of the key neurons which are involved in these two things, may contribute to the pathogenesis of obesity and diabetes, and that, again, being able to understand the select neurons, how they're activated and how they're regulated, if we are being able to target those particular neurons, is that there may be sort of a therapeutic treatment, not only for obesity, but also diabetes as well. Finally, thank to those people who sort of worked with me through this. I mentored Mike Schwartz, I mentored work from Jared, other members in the lab, both past and present, particularly the work of Jennifer Deem and Thomas Meek, those at the University of Washington, Richard Parmenter for providing the mouse model, Michael Brukus to help with the fibre photometry and funding. So thank you very much for your attention. I'd be more than happy to answer any questions.
Thank you, Dr. Morton. We do have mic runners in the room, and we will look for any questions. Excellent. I'll give you my mic.
Doctor, I was wondering about whether or not chronologically, as you were describing the AGRP studies on the AGRP neurons, was the fat studied first as an energy molecule, followed by the glucose studies? Was that a necessary order? Was that done for a specific reason?
Yeah, so again, part of it in terms of the energy homeostasis field, it was probably sort of more well established to begin with. And again, the challenge in the field really has been is in terms of any effects being able to just disassociate the effect of weight versus blood sugar. Because we all know is that the best way to be able to help control your diabetes is to lose a few pounds. So how do you sort of separate those effects apart? It becomes more confusing, obviously, is that not only the same brain regions are involved, but some of the same neurons are involved as well. What we haven't got to the stage yet is, again, the hypothesis is that some neurons are selectively regulating the feeding pathways and other neurons are selectively regulating the blood sugar pathways. That's the stage that we're at now. So in terms of the diabetes field, it's been a bit further behind because, again, most of the work has all been focused on insulin secretion. You have the liver people say everything's about the liver. You have the muscle people say, no, insulin resistance is all about the muscle. So the brain was really forgotten. Now, what we're interested in terms of is like, not that the brain can do something. So obviously that's been the first start that we've done, sort of using more pharmacology. But what we're really trying to understand, what is the physiological of the brain in sort of day in, day out, meal to meal interactions. Quite often at the moment still is that we're sort of, in an intact system, the brain, we hypothesize, is just monitoring things. If everything's good, it doesn't really need to pull any, strings at all. But when all of a sudden you get challenged under different circumstances, obviously cold exposure as well, because not only in terms of the increase of energy expenditure is associated, that takes up a large amount of fuel to be able to do that. If all that fuel is going to thermogenesis, you're at risk of being able to become hypoglycemic. So the brain then actually helps control things to be able to make sure the blood sugars stay similar. The other thing too is that probably the other best evidence is in terms of the response to hypoglycemia. Because there, obviously, if you're giving insulin exogenously, is that, and you sort of have that lower blood sugar, is the brain will completely take control over the beta cell in those circumstances. So obviously then it's like an emergency system. The brain will completely shut off the islet in terms of the beta cells. And then it has powerful mechanisms that it can engage to be able to rise the blood sugars, back up. This is obviously independent of food intake. And obviously part of that's through the autonomic nervous system in terms of effects to be able to, particularly on the liver, be able to try and sort of, drive glucose out of the liver to be able to sort of bring the level back up to normal.
Thank you.
All right, now we're in business. Thank you, Dr. Morton. I'm just curious, and I'll admit I'm just tipping my toe into this research, but a big thing that we're also seeing is the link on A1C levels and mood. And with your work, if you're seeing anything with like neurotransmitters, because one thing I'm thinking of is in Alaska, we have a lot of cold. We also have a lot of mental health concerns and a lot of comorbidities that are probably also impacting our customer owners, our communities as they're addressing obesity too. So I know that's like a big question with 2 minutes to go, but if you've seen like just some links in your research, I'd love to hear. Thank you.
Yeah, that's sort of the moving frontier in terms of field. Again, most of the field, is focused on the hypothalamus in terms of homeostatic system. But now the field is moving forward out of that, and a lot of it in terms we don't just eat for homeostasis, and there's the reward and there's the motivation side of things. And there's other aspects of looking in terms of cognition and so forth. And so obviously you can also put sort of mood into that as well. And so we're really only getting the tip of the iceberg in terms of sort of understanding this. And obviously part of that is in terms of some of the new therapies that are coming out in terms of really impressive weight loss with the class of GLP-1 receptor antagonists, is that for some of these things is that we don't know the full mechanism of action. Yes, we know in terms of the satiety effects and can explain some of the nausea, but as we heard today, there's also reduced cravings. There's not only to food, but alcohol and other things. They're starting to look at in terms of other drug addictions, and it's like, well, GLP-1 receptors, some are known to be expressed in those areas. Are they mediating those particular effects? And so I think we're going to start to move further and further into that field to be able to sort of look at the integration of these different brain centers.
Thanks for a really talk. You've alluded to this a couple of times during the talk. There's sort of a correlate compartment, hypothetical, of course, that may be totally separate from this, and involves the etiology of insulin resistance, the effect on GLUT transport, glucose disposal in the muscle and the liver, et cetera. Do you anticipate a discovery of a nexus that might unify those two hypothetical compartments that lead ultimately to hyperglycemia, especially in type 2 diabetes?
Are you talking about in terms of effect on the periphery or the brain or both?
Both.
Yeah, that's something that we've really sort of, thought long and hard about in terms of, is there an integration point in it? Because again, as we said, is that in terms of what is a real link with obesity, insulin resistance, but also more so in terms of diabetes as well. Is there a sort of a common pathway that sort of ties those together to be able to help explain in terms of some individuals, obese but don't become diabetic, in terms of those that do become diabetic? Obviously, in terms of that integration response, we're obviously interested in this particular set of neurons, those that express the AGRP, whether they actually are sort of associated or not. In terms of both obesity, there is evidence in probably the most common forms of genetic forms of obesity, sort of mutations in the melanocortin pathway and those receptors. At this stage, I haven't necessarily looked to be able to see if those associations also hold true in terms of those with diabetes. But that could be one potentially sort of key sort of area which sort of overlaps with the time with both obesity and diabetes.
All right, thank you, Dr. Morton. Will you be, yep.